So feedback is typically, it does involve signaling, which is why it's in this cells and cell signaling unit. Because basically it's how the cells know like when to stop a reaction or when to start one. And that's based on, you know, signals that they get from outside. However, it's kind of loosely connected. So really what we have are two different types of feedback. And feedback is typically something that where we're looking at trying to maintain a balance in the body. And so this is for a multicellular organism for the most part. Although we can see feedback in unicellular, but I'm going to deal with it first with just like talking about us, okay? Because it's a little bit easier to understand. So there's two different types of feedback. Negative feedback is the one that we see most consistently in our like day-to-day life. And that's because it's maintaining homeostasis. Now, if you don't know what homeostasis is because you don't remember, that's basically a term that means like a balance in an organism. So your body has to be balanced. Every organism has to have some sort of balance, or else, of course, we know we have to live within an optimal range, like of temperature and pH and all of that, water, or else we'll die. And so that's kind of where homeostasis comes in. Like we have to have like a set point that like this is optimal, and outside of this range, systems start to fail. And so the one I've put on here, this is probably the most easy to understand negative feedback example. And remember, that is to maintain homeostasis. So we're trying to stay like in the middle, like at a set point, is our body temperature. And so we all know that we maintain our own body temperature. We're an organism that can do that. But if we have different strategies. Our body has different strategies whenever something changes our body temperature. So like if our body temperature increases, like it shows this little guy here, he's turning red because he's increasing in temperature. So if our body temperature increases, then we'll start having some cooling mechanisms. So sweating is one of the main ones that we see, but also we can see things like inside, things will happen inside that we don't really know about, like vasodilation, like our veins will get bigger because anytime something gets bigger, it gives it more surface area, and then there's more coolant space, stuff like that. So we might, we have sweating actually wicks heat out and then evaporates it away so that it takes heat out of your body. Elephants have these really big ears. That's all for cooling. And they'll start kind of flapping their ears so that they can cool the blood in their ears so that it's cooler in their body. But those are all strategies to decrease then the body temperature back to the set point, okay? However, if it starts to cool too much, so like you're sweating too much, the elephants are flapping their ears too much, whatever happens too much, then you start to have heating mechanisms activated. So you start to shiver, your veins constrict. All these different things go into effect that make you begin to warm back up. And so then it just continues. This is a negative feedback, meaning if you have input into the system, your body will react to bring it back down. Or if you go below, then your body will react to bring it back up. So we call that a negative feedback loop, basically just maintaining homeostasis. Any action will have a negative action against it. So if you see an increase in temperature, you'll have a decrease mechanism, and vice versa. If you have a decrease, then you'll have increase. So it's like every action that happens has a negative reaction against it to maintain homeostasis in whatever organism it is. Okay. How are we on negative feedback? Totally makes sense. Like it's not a difficult concept. You just have to know the vocab. All right. So then we have to have some positive feedback. So this one I've switched. I didn't do the human one. I'll bring it up later. This one's actually the one that's most typically used in AP Bio. So I wanted to use these examples first. Positive feedback is completely different. And a positive feedback, you're wanting to have some result that's not a typical day-to-day result. And it needs to happen. So it's not trying to keep it in a balance. Like you're actually trying to knock it out of balance so you can amplify something. And it'll build and build and build and build until the event happens, and then everything crashes down and stops. Okay, so this is a positive feedback. It's like when you say temperature was positive feedback. So it'd be like when I increase temperature, then I'm also going to do the shivering. So I'll increase it some more until it's like, I don't know, until you blow up or something. But that's not what temperature is. Okay, that temperature is negative because we need to keep it balanced. These are events that you want to build until you get some something done, some result. And so it took a little bit harder to define. So for positive feedback, I put we're creating or changing some result. Like we're looking for some result that's not a typical homeostasis thing. And you can usually see these because they're amplifying something, like something is getting bigger and bigger and bigger, or smaller and smaller and smaller and smaller. Okay, so some reaction within. So let's see where we're going to start this. So we have fruit begins to ripen, and you may have noticed this. I don't know if you have or not. If you have like a bag of apples that you bought at the store, you might have like one apple that's kind of more ripe than the others, and then all of a sudden like the whole bag starts to ripen and they get all nasty at the same time, and you're like, "Ooh, what just happened? Like my apples were fine yesterday and now they're like mushy." So fruit actually ripening fruit releases a gas called ethylene. Ethylene is it's just naturally a hormone that's released into the air. So it's like a vaporized hormone that's released into the air for ripening fruit. So what does this do for the organism? Well, when they have one apple ripen on the tree, it'll release some ethylene because it's just a typical ripening hormone. Like in the process of ripening, ethylene is produced and released. When that happens, then it actually triggers it. Oh, now my little bar popped up and you can't see it. It triggers ethylene to increase, which makes nearby fruit ripen. And then it actually starts right here. Ethylene produced in one fruit and then is detected by nearby fruit, and then nearby fruit begins to ripen and release ethylene, and then ethylene increases, and then more fruits ripen, and then blah, 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 on and on and on until all the apples are ripe on the whole entire tree. And really why that would be beneficial, like an evolutionary speaking for the plant, is that fruit's job is to distribute seeds. And so if an apple falls off the tree, or if it's ripe and something eats it, then they'll walk off somewhere and poop out the seeds somewhere, and it's like built-in fertilizer in the seeds. That's the role of fruit. That's why it's sweet and juicy and attractant to things. So it's best for the plant if it can get all of its seeds ripen at once to attract a lot of seed dispersal organisms, so that it can have more of a chance of them, being dispersed. And so this is what they do, a positive feedback, where you just have more and more stuff produced until boom, they are all ripe at the same time. And you have like a harvest, and all the little organisms come and eat them and go poop the seeds out. And next spring, you got new trees grown. Okay, questions about positive feedback? Everybody good? Okay. So now some examples. Now you're going to tell me if this is a positive or a negative feedback cycle. So this could be definitely like an AP question where they have an example on there, and you're supposed to identify what type of feedback we're looking at. So it looks like they put numbers on them so that you can tell where to start. Sometimes it's hard to tell where to start. So it looks like you've had an injury, a break or tear in a blood vessel or wall. And then clotting occurs as platelets adhere and release chemicals. So the platelets are releasing, signaling out, saying, woo, we need help. So then clotting proceeds until the break is sealed by newly formed clot. Released chemicals attract more platelets, and then this is good. Now, of course, we're not going to have any more breaking or any more of that. I mean, it's like these two things are like receiving the help from the platelets. So we're going to stick with the platelets. So then it attracts more platelets, and then they adhere, and then they attract more platelets, and then they adhere. What kind of feedback is this?
Would that be negative?
This one's actually positive. 50-50 chance, dang it. This one's positive because it will continue to do this, build and build and build and build until it's sealed completely, and then it'll just stop dead, right? Does that make sense? So it's not like it's saying, oh, we've built up too many platelets, now we have to break down platelets. Oh, now we have to build up platelets, now we have to break down platelets. Like, it's not trying to find some happy medium. It's trying to build and build and build and build clots until the wound is sealed, and then all the signaling will just magically stop. Like the cells they'll be basically, whenever they butt really super close together, they start to like stop. It begins to stop the signaling. And so when the signaling stops, it's like done. The clot is finished, and we don't need to do that anymore. And so all the signaling stops. That's positive feedback. Okay. Built and built and built and built until the job was done, and then check, done.
Okay,
Good.
I was thinking negative because it ends where it started, basically.
So yeah, and most of them will look like a cycle like this, you know, kind of like they look like a circle, like all diagrams for feedback look like a circle. It's kind of weird. Negative will often have 2 loops, and I'll show you a negative double loop circle. But positive is just going like it's going round and round and round and round and round until it's like finish the job. And so that's what you're looking for in a positive feedback, is you're looking for like a job that needs to be finished. And for a negative feedback, you're looking for like an ongoing, like day, every day kind of balance. That's what we're looking for. Okay, let's look at this one. All right, we are looking to stimulate spermatogenesis, which means we're going to send something through meiosis to make sperm. And it looks like we've got some hormones going on here. So I just stuck this one on here because it has so many weird terms you've never heard of before. Because that's always nice to look at diagrams whenever you're like, I don't even know what any of these words mean, because you'll get that on the AP test. So it looks like we have a pituitary, or a hypothalamus is in your brain, and it looks like it's stimulating the anterior pituitary, which is also in your brain. And it looks like that is releasing something, FSH and LH. And I mean, I know what those are, but you don't need to, so that's fine. And so then it looks like we're making sperm here, and there's something to do with these Leydig cells, but we don't care what they are. And it looks like the Leydig cells are causing more sperm to be made, okay? But then looks like what sperm is starting to make, we're inhibiting it so that we don't have like a bunch more made. And then it sees, oh, we don't have enough sperm. And so then it triggers that to create some more. And then the actual production of the sperm like turns that off. And then, oh, not enough sperm. Okay, what do we got going on here? Positive or negative?
Negative feedback.
Negative feedback, because we're keeping a balance. Like we don't want too many, but then we don't want none either. Like we want the right amount. And so it's like the actual production of the sperm is inhibiting more being produced until the anterior pituitary is like, wow, we don't have any. And so then it goes again. Okay, so this is negative because it's trying to keep a balance. It's not like building and building and building and then stop sperm production forever. It's like just keeping kind of a mid-range. Okay. Okay, so now we have a baby being born. Wow, all this reproduction stuff, I promise there's only 2. So now we have a baby being born, and this might give some people the heebie-jeebies, but it's okay. So here's baby. She or he or whatever is in the uterus, and it's pressing on the cervix, which is the opening of the uterus, where usually this would be like where the baby's going to be born. So it's going to move out birth canal and out of the mom. And so basically we've got some really interesting stuff that goes on here, but this is super simplified, which is nice. So the baby pushes against the cervix. So its head pushes against the opening of the uterus. And then that stretching of the cervix causes nerve impulses to be sent to the brain. And then the brain stimulates posterior pituitary to release oxytocin. And then oxytocin causes the muscles in the uterus to contract, so to squeeze the baby, which causes the baby's head to push against the cervix, which causes stretching of the cervix, which causes the brain to release more oxytocin, which causes the, what kind of feedback is this?
Is that positive?
Positive, because that will continue to increase until the baby's born. And once the baby's born, it'll just shut off. Okay, it's got a task to do. That is not a typical reaction. It's not a day-to-day, daily thing, right? It is a task to do, and it will increase until the task is done. And then the signaling will shut off. Questions? Okay, let's look at another one. Aha, pH of your blood. So your normal blood is 7, a little bit more, okay? A little bit higher than 7, but quite neutral. And so if you have an increase in pH, then your chemoreceptors in your kidneys will notice it, and then they will secrete hydrogen ions, which will cause your pH to drop. If they secrete too much hydrogen, see, increase in hydrogen ions, pH drops too low, then chemoreceptors in kidneys will signal the medulla oblongata, which is in your brain, and chemoreceptors in the brain will signal the kidneys, which will then stop the release of H, and in fact can release OH, which will then bring it back to normal, unless it goes too much. And then if it goes too much, then he goes, okay, what kind of feedback is this?
Negative.
Negative feedback, right? We are trying to keep this normal, which if you ever hear of diets and stuff that are supposed to change your blood pH, just remember this diagram, because your blood pH is held super neutral, and it's really difficult to change your body's pH. Like if you start taking apple cider vinegar thinking you're going to like decrease your pH of your body, your kidneys are just going to release more OH and thwart all your efforts to change your blood pH.
Ms. Swoy,
Yep.
so if a diagram shows that you're trying to maintain homeostasis, then can you assume that it's always going to be negative then?
Yes, because positive feedback isn't maintaining homeostasis. It's got like a task to complete. It's actually knocking the body out of homeostasis briefly. Okay. Oh, looks like we have a loop. So what's this probably going to be? Because we have a set point right here in the middle.
Positive.
Negative. This is negative. Yep, negative because we have a normal blood calcium level, and then you can see it like increasing and doing stuff and bringing it back down to normal. And then you can see it, oh gosh, decreasing, and then, oh, doing some stuff and bringing it back up to normal. You have a set point of normal blood calcium level, and your body's going to react to a rising blood calcium this way and bring it back down. Or if you have falling, it'll bring it back up. Now, so, guys, you're kind of stuck in a loop your whole entire life of this. Like you don't even know this is going on. But if you overreact to your calcium level, then and it goes too far down, then your body will fix it, and vice versa. And it's just, this is just happening inside your body all the time. Like your body's releasing chemicals and being like, whoa, we're kind of out of whack. Let's, bring it back up. Too much. Let's bring it back down. And it's just like this very, delicate balance. So that's a negative feedback. It has a set point it's trying to maintain. Okay. What about operons? This is a good time to talk about operons because we have tests coming up and operons are going to be on it. So this is just any old operon, could be anything. The thing about operons is, remember, this is where and this is in bacteria usually. So this is single cell. So this isn't, this isn't the whole body systems we've been looking at. So basically what we've got is we have a gene that is repressed. So it's got a repressor on it. It's just sitting there. If it's repressed all the time, waiting for a signal, is that inducible or repressible?
Inducible?
Inducible. What you're going to say? Yeah, it's inducible. So this is an inducible operon, just waiting for a signal. And so what's going to happen then is when something builds up, you see the little somethings building up here, then it'll lock in, and then this will produce whatever. Like I said, this is just generic, so it could be anything. This will produce whatever until all of these little things are used to the point where like that one is used as well. Okay, and since you're trying to maintain, this is your like normal, this is your like kind of homeostasis set point. Whenever that is all used up, then it'll lock back on. Then we would consider operons a negative feedback because what they are producing is regulating themselves. Okay, and so if they produce a lot of glucose, say, from this, like, okay, let's just say this is a lac operon. This is a lac operon, and this is lactose sugar. Okay, and it's saying, oh gosh, there's lactose here. We need to get the repressor off, right? And so that's what happened. Well, lactose is broken down into glucose and galactose. So the system only needs glucose. Once glucose is present, then this system shuts off. And so basically it has regulated itself by turning on when there was lactose present, and then turning off when it's like broken up enough lactose to continue, like doesn't need to do anymore. So it's self-regulating. Basically, its own products shut it down. That's why all operons basically run on as a negative feedback, because their own products either shut them down or their own products can turn them back on if it's a repressible. Okay, does that make sense? Whenever it's self-regulating, maintaining homeostasis, then we consider that negative feedback. Okay, any questions? That was pretty simple. I gave you a little like 20-minute lab activity to do in Canvas, and it's in Pivot. And I broke it, I pared it down to just one section, and it's about regulating glucose. And I even took off the graphing portion. So all you have to do is answer questions, and you do have to collect some data to just see what's happening with a diabetic person versus a non-diabetic person whenever they are eating. and what happens to their blood glucose levels. And then there's a feedback loop, and you're supposed to identify it and all that stuff. But let me know if it's taking you any more than 20, 25 minutes. Like it shouldn't. I think I pared it down to like, I don't know, 15 questions or something like that total. Okay.
If an operon is repressible, what does it look like?
It would look like this bottom one. And then, except for there wouldn't be anything locked in. And when that thing locked into it, it would sit down.
Oh, okay, okay. But
so when it was
hovering
down,
around
it would,
the DNA thing.
Yeah, it would just be hovering around the gene. Yeah, the DNA,
The gene.
waiting. And then basically what it is the protein doesn't fit unless it has something in its active site. So the protein wouldn't fit on this particular little thing unless it had something in its active site that was modifying its shape.
Okay.
So once the active,
what is number 4 called again? The 4 thing?
oh, this thing? This is the operon. That's the operator region.
Is the operon?
Sorry, the operator region.
Oh, the operator region. Okay, cool, cool.
And that's why it's called an operon, because it has an operator. Okay. And this is the promoter region, number 3, operator. And then these are your structural genes that are going to be produced if the yellow thing, the polymerase, can get across. Okay. All right. Any other questions?